Okay. Good afternoon, students. Can you tell me, guys, whether I'm audible and my screen is visible so that I will start the class, okay? Yeah, can you tell me whether I'm audible and my screen is visible? Yeah, thank you so much. Okay. So let us start the class, guys. In the last class, we have, yeah, thank one. Okay. In the last class, we have started with the sexual reproduction of the flowering plant, right? In the last class, we have did the artificial hybridization. Before the artificial hybridization, we have completed the microsporogenesis, right? We have completed microsporogenesis. That is nothing but the formation of the pollen grain we have studied, right? Formation of the pollen grain. Then megasporogenesis we studied. Megasporogenesis. Megasporogenesis is formation of the embryo sac, right, guys? In the female, that is embryo sac formation in the ovule, right? Then we studied out the pollen and pistil interaction, then pollination, right? And in the last class, we studied about the artificial hybridization. In a today's class, we are going to continue the double fertilization. Okay. So before going to start the class, guys, here is a question for you. The question is attractant and the reward. Okay. Attractant and the reward are required for which type of the pollination? Option A, entomophily. Option B, hydrophily. Option C, clistogamy. Option D, anemophily. Okay. You can answer me in the chat box also, and meanwhile, I will run the poll also. You can answer me in the poll. Okay. I want everyone to answer to this question. Yeah. Had your lunch, guys? You all are not sleepy, right? Then I want answer from everyone. Very good, Monica. Terikam, can you think once again? Very good, Sanjushri. Clarkson, very good. Okay. So in the last class, we studied, right, guys, in the pollination, the attractants and the rewards. We have to give attractants and the reward for the wind. Think once again. The pollination by the wind is called as anemophily, right? For the wind, are we going to give any reward? If that wind is going to take any, it is going to be take any reward, guys? No, right? No, right? Yes, HP. Yes, Arun. So the correct answer is A. Right? Most of them given A answer, but most of them given 11% C, D, and B. All are given wrong answer. Read once again the topic. Okay. So what we studied, guys, the attractants and the rewards we require for the insect pollination because it has to attract the insect, right? For the a bright color, for the colored petals, the insect has to be attracted toward the flower, right? For that, we have to give a attractant, right? Then we have to give a nectar as a reward, right? We have to, that flower has to give a nectar as a reward. For what? Is that wind is going to take any nectar? No, right? Entomophily. Entomophily means insect pollination. For the insect pollination, we require the attractant and the reward. Hydrophily, no. For this, we don't require. Whereas a clistogamy is nothing but a closed flower, right? In the closed flower, there is no cross pollination. So that's why we don't require any agent. In this, only the autogamy is going to be happen. Autogamy, right? In the closed flower, there is no cross pollination, only self pollination. Okay. Then coming to the anemophily, the pollination by the wind. Pollination by the wind is called as anemophily. Okay. So if you understood this question means kindly give me a thumbs up or your emojis. Okay. I'm expecting your emojis, guys, in the chat box. Okay, fine. So let us go to the next question. The flower which have a single ovule in the ovary and are packed into inflorescence. Okay, so these flowers are packed into the inflorescence. Okay, these are packed into the inflorescence, but in this it is having only one ovule. It is having only one ovule means, tell me which type of the pollination it is. O is going to be do the pollination here. B, wind, bat, water. Thank you. Very, Serikan, think once again. This is actually, you have to remember, guys, this is like a unique character. Unique character, this is. You have to remember, very good, Jirmili. Very good, Harman. Very good, Carlson. Yeah. So I will launch the poll, guys. Let us see how many of you will answer to this. Yeah, I'm relaunching the poll. You can give me your answers, guys. Very good, Nikita. Very good, Sanju. Very good, Navya. Navya, you're from which JNV? Very good, guys. Very good. Most of them is given the correct answer, that is B, and some given a C because they are getting confused. Okay, no problem. See here, guys. The flower which have a single ovule, this is a unique character. Just remember, okay. Single ovule in the ovary and are packed into inflorescence. This is the characteristics of wind pollination. Wind. Okay. Fine. Let us come to the bee. For the bee pollination, we didn't study these characters, right? That is a single flower only it is there, not the inflorescence, right? Okay. And you know that for the bat, we require the a large flower. It is not packed into the inflorescence, right? That is a large flower required for the bat and it should be a dull colored, right? Okay. Then I think you understood about this. Let us go to the double fertilization. So first, let us learn what is called as double fertilization. Okay, kindly go through this statement, guys. Meanwhile, I will explain you. A double fertilization is the fusion of two male gamete. You know that two male gamete is nothing but the generative cell. Always I will tell generative cell, right? Generative cell brought by the pollen tube to the brought by the pollen tube to two different cell. For the two different cell, the pollen tube is going to be 1 minute. One minute, guys. Yeah. Here you can see that the pollen tube is bringing the two male gamete, right? The two male gamete for the fusion of the two different cell. For the two different cell and for the same female gametophyte. For the same female gametophyte. In order to produce two different structure. Here what is happening? The pollen tube is bringing the two male gamete for the two different cell. Which are the two different cell here? One is egg cell and another one is a central cell, right? It is bringing the male gamete to the one to the egg cell and another to the central cell. Two different cell, right? Now, this is going to form the two different structure. Which are the two different structure? Embryo, the egg cell fusing, it is going to form the embryo, and the fusion with the central cell, it is going to form the endosperm. Endosperm, right? So that's why it is called as double fertilization. Kindly read once again. The double fertilization is the fusion of two male gametes brought by the pollen tube to two different cell, egg cell and the central cell of the same female gametophyte in order to produce two different structure. Two different structure is nothing but embryo and the endosperm. Okay. So that is called as double fertilization. And double fertilization is a characteristic feature of angiosperm. We can see only in the angiosperm. And it is discovered by Navasin in 1898. Okay. Yes, Navyashri, I didn't on the video because network issue is there here. That's why, Pa. My screen is visible, right? Navya, my screen PPT is visible, right? Yes, okay, Pa. Only for today, okay? Fine. Then coming to the pollen tube. So here is the scientist name, okay? Now let us talk about the pollen tube. So you all know that pollen tube will enter the embryo sac by the three ways, right? Now I need the answer from you. I want my chat box to fill with your answer, okay? So now you tell me quickly, if the pollen tube is entering through the chalazal end, what is this called as? Chalazal end. If the pollen tube entering through the chalazal end, it is called as, what is that called as? Chalazogamy. Very good, Venu. If it is entering through the micropylar end, it is called as. Only Venu is answering. What about others? What about Amiya, Adi, G, Gauri Lakshmi, and Arun, HP, Mahalakshmi? Answer me. Porogamy. Very good, Venu. Only Venu is there in the class. Very good, Larti. Very good. Porogamy. And what about if it is entering through the center? Integuments. To the integument means it is called as mesogamy. Very good. Very good, Venu. Okay. So now the pollen tube entered, right? When the pollen tube entered, it is going to be burst open. Okay. The pollen tube has to be entered, right? Now the pollen tube entered. See here, the entered into the ovule. There is a egg cell is there and synergid cells are there. Right. This is the egg cell. Observe here. This is the egg cell and these are the synergids. Right. Now what will happen? This pollen tube will enter to this synergid and there it is going to be burst the synergid. Okay. The pollen tube is going to be burst out the synergid and it is going to release its two gametes. Okay. When this release its two gametes, one gamete is going to be fused with the central cell. Okay. This gamete is going to fuse with the central cell and this is going to be from the primary endosperm nucleus. Okay. And the another gamete is going to fuse with the egg cell. Egg cell. Okay, so that is I have given here. The pollen tube burst open in one of the two synergids and release the two male gamete. Now, one of the male gamete, it is fused with the egg. Now, the male gamete, one of the male gamete, it is fused with this egg, right? Now, it is going to be from the zygote. You all know that zygote is a diploid in nature. Yes or no? Yes. Then, this fertilization, the egg cell that is combining with the one of the male gamete, that is forming the zygote. It is going to form the main zygote. That called as a generative fertilization or true fertilization. Kindly remember this. It is called as true fertilization and generative fertilization. Okay. Next, what will happen? The second male gamete is there, right? The second male gamete now fuse with the diploid secondary nucleus. See here, it is called as a diploid. Why we are going to call as? Remember, in the most of the exams, you will get the question. Central cell. What is the ploidy of central cell? Tell me, guys, what is the ploidy of central cell? Can I expect the answer from you guys? What is the ploidy of central cell? Yeah, what is the ploidy of central cell? 2n. Very good, Venu. Very good. 2n. Right. What about the others, guys? Very good, Mahalakshmi. Yes, 2n. Yes. Yes, Carlson. Yes, Navyashri. Very good. Now what will happen? One of the male gamete is going to fuse with this. 2n means what will happen now? It is going to be formed. It is going to be formed. 3n. Right. Now, one of the male gamete is very good, Nikita. Very good, Larti. If it is going to fuse with this 2n central cell means a diploid central cell means Now the ploidy will become 3n. Now, right? Now the ploidy become 3n, right? Now the central cell to form the triploid primary endosperm, that is called as pen. Pen. Okay, in the most of the exam you will get the question that pen is diploid or haploid or triploid. That is triploid, 3n. Okay. These fertilization, this fertilization we are going to call as vegetative fertilization. Why we are going to call as a vegetative fertilization means it is going to form the endosperm. Remember guys, this 3n fertilization is going to be form the endosperm, whereas this is going to produce this is going to give the nourishment to the growing embryo. This endosperm is going to give a nourishment or the food for the growing embryo. Remember, food for embryo, growing embryo. So that's why it is called as vegetative fertilization. Okay, fine. So the endosperm will form after the formation of the embryo. It is very, very important. What will happen here in the angiosperm is, in the angiosperm, first the embryo will form. Okay. The fertilization will happen. The embryo will form. After the embryo, after the formation of the embryo, immediately the endosperm is going to be formed. Why actually? If the endosperm is formed before the fertilization or the before the formation of the embryo means there will be a, if the fertilization is not happened means the energy given for making the endosperm is wasted, right? So that's why in the angiosperm mainly the first endosperm is going to be formed. Endosperm is going to be formed. Sorry, the first embryo is going to be formed in the angiosperm. Later the endosperm is going to be formed. But in the gymnosperm, remember, in the gymnosperm it is vice versa. Okay. In the gymnosperm, in the gymnosperm, okay, remember here, in the gymnosperm, the first will be endosperm formation will happen, endosperm, whereas the second embryo. Kindly make a note of this, guys. Here I don't have a place, that's why I'm writing here. In the angiosperm, the first embryo, then endosperm. But in the gymnosperm, the first endosperm will form, in the second embryo will form. Okay, fine. I think you understood this, what are the things I told you, and you know that the endosperm will provide the nutrition for growing embryo. Okay. Kindly see this video, guys. I am playing here this video. How the pollen tube is going to be grow and how this pollen tube is enters the ovule and fusing with the egg and the central cell. Kindly watch this video. Pollen grain, stigma. Now pollen tube is forming. Now it is entering through the micropylar end. Then one gamete, it is going to fuse with the central cell and another with the egg cell. So this is what we are going to call as double fertilization. Uh, yes, sir. Not mainly, Nikita. Some plants we will use for the decorative purpose, but there will be a no flower will be there, right? Sorry. Only some plants we will use it for the decorative purpose. Thank you. But that thing decorative purpose and here it is not connected. Okay. Do you understood double fertilization, guys? Do you have any question in the double fertilization? Do you understood clearly? Give me thumbs up or emojis. Kindly make a notes also. If I didn't get any emojis, what can I understood, guys? Now only you had lunch. I should think all are sleeping. What about Shiva, Venu, Vedashri, Tina? Very good, Monica. Very good, HP. For HP, I am asking the name from past class. HP is not telling which what is the name and from which JNV. Is that HP is any JNV or your student from any JNV HP? Because name is not there, no? That's why I'm asking. Yes, Ranganayaki. Okay, fine. I think you understood this. Here you can see, guys. This is the same diagram here. I've explained you last class also. If you take this is as a stigma, this is a stigma. That is this is a pistil. On the stigma, the pollen is receiving. Here you can see that in the ovary there will be so many ovules are there. Okay, so many ovules are there. I'm revising once again. All are not understood. Kindly see here, right there. Okay. And in this ovule we are taking, we are zooming out. Okay, this ovule, the pollen tube is entered, right? This pollen tube is entered to this ovule, and we are zooming this ovule. Now the pollen tube will burst out the synergid cell, and it is going to be released the two gamete. One gamete is going to be fused with the central cell, and one is with the egg cell. Okay, one is with the egg cell. Now the central cell is going to be from the pen, that is primary endosperm nucleus, and the one nucleus which is fused with the egg, that is going to be from the zygote. Now zygote is going to form the future embryo, and this pen will form the endosperm. Endosperm. Okay, this is about the double fertilization. Okay, so this is what we already studied. This is in the stepwise manner it is there. You can go through this. Okay, so here you can see. I have told you some some angiosperm will shed at the two-cell stage. Two-cell stage, right? So here only one generative cell is there and one tube nucleus, that is nothing but the vegetative cell nucleus is there, right? So what will happen? Vegetative cell nucleus is there, right? So here the generative cell is going to be divided mitotically. Okay. Now it is going to form the two male gamete. And it is fusing. This is the central cell and with the egg. Okay. Here you can see in the fourth point, the synergids will break down. One sperm nucleus unites with the two polar nuclei, forming the first cell of the 3N endosperm generation. And another with the nucleus with the egg. Okay, fine, guys. So I think we understood this. No need to explain this. So if you not understood clear limits, you can make this diagram. You can make this stepwise thing. One male gamete, it is fusing with the egg. And it is going to be formed a diploid because egg is also haploid and the, male gamete is also haploid and the egg is also haploid. So that's why it is going to form the diploid. This is the embryo. Then second male gamete, that is going to be fused with the diploid central cell, and the male gamete is also haploid. With together with this 2N, it is going to be form of the 3N, and it is going to be form a endosperm. You can see here, guys. This is the embryo. Center wala you can see that is the embryo, and the surrounding, that is the endosperm, that is giving the nutrition. Okay. One minute. And here is the question for you guys. If you understood this means, you can tell me the answer. The triple fusion involves egg and two synergids, two polar nuclei and the male gamete, one polar nuclei, egg and male gamete, egg and two antipodal cell. The triple fusion involves triple fusion. We are talking about triple fusion mainly. The formation of the pen. Formation of the pen is also called as triple fusion. Very good, very good. Very good, Sanjushri. Very good, Tengwan. Very good, Dharmi. Very good, Navya. Yes. So let us talk about the answer, guys. Answer is B, right? Here what is happening? The male gamete is going to be fused with the central cell, right? Central cell. The male gamete will fuse, right? This male gamete will fuse. This is also called as triple fusion. Triple fusion. In the triple fusion, which all the thing is involved? One is male gamete. Male gamete. And the second one is two polar nuclei, that is in the central cell. Two polar nuclei are in the central cell, right? So that's why we are going to tell that two polar nuclei. Two polar nuclei. Did you understood this? Okay, let us talk about after the fertilization, what are the things is going to be happen. Before that also we'll solve some questions. Yeah. So here is a question for you guys. Double fertilization is a characteristic of. Double fertilization is a characteristic of. Option A, flowering plant. Kindly see question number 1 only. Okay. And option B, cycas. Option C, pinus. And option D, gymnosperm. Very good, Venu. Very good, Vana. Navyashri. Very good, HP. Very good, Ranganayaki. Very good, Lakshmi Kumar. Very good, Nikita. Very good. G. Very good, Tangwan. See here, the double fertilization, now only we studied that is the characteristic of the flowering plant, right? The correct answer is A. The cycas and pinus both will come under the gymnosperm. Okay, remember, both are gymnosperm only. Cycas and the pinus. Okay. Next, second question. Kindly see the second question. The process of removing the stamen from the flower is called as stamen from the flower. In the last class, we studied artificial hybridization. Very good, very good. JNV Hyderabad, Tanuja, Sanjushri, Ranganayaki. Very good. The correct answer is artificial. What is that correct answer? In the artificial hybridization, we will see that removal of the stamen we call as emasculation. And bagging of the bagging of the stigma by the butter paper and the polythene is called as a bagging, right? So the correct answer is emasculation, option C. Then third one, outbreeding devices prevent. Why the plants cultivated the outbreeding devices? Option A, variation. Option B, inbreeding depression. Option C, autogamy. Option D, both B and C. Think once again, guys. What will happen in the, we studied the disadvantages of the self-pollination or autogamy, right? What is the disadvantages or the, what is the disadvantages of self-pollination or the autogamy? That is also comes under this question. Very good, Venu. Very good, Nikita. Ranganayaki Bhargavi, think once again, pa. Very good, Ranganayaki. Yes. Yes, Sanju. Correct. Very good, Malakshmi. Yeah. During the self-pollination, if the self-pollination is goes on increasing, means if the plants is undergoing the self-pollination only means, what is going to be happen? Inbreeding depression, right? There will be a no variation will be there, right? There will be a inbreeding depression is going to be happen in the self-pollination, Okay. So that is also correct. And the autogamy is nothing but self-pollination only. So that's why B and C is the correct answer. So D is the correct answer, both B and C. Okay, fine. So now we are completed with now we are all completed with the one minute. Yeah. Now we all completed with the fertilization, right? The fertilization is completed now. Now the task is after the fertilization, what is going to be happen, guys? After the fertilization, what is the task? Sanjushri, have you raised the hand, pa? Sanjushri, do you have any question? You have raised your hand. Sanjushri? Okay, by miss. Okay. After the fertilization, the endosperm is there, right? It is going to be developed. Okay, the endosperm development is going to be happen. Kindly make a note of this. Endosperm is going to be developed, and the embryo, the zygote is going to be developed into the embryo. Okay, and the ovule is there, right? Inside the ovule, embryo is there. Now, ovule is going to be converted into the seed. Ovule, you all know that inside the ovule, the embryo sac is there. Embryo sac. right? Embryo sac is there. Here the egg is fused and endosperm is forming. This is going to be formed the embryo means the outside that there is a ovule is there. That ovule is going to be formed the seed. The seed you can see, right? Seeds. Now the ovary, you know that where is the ovule is present? Inside the ovary, right? Inside the ovary, ovule is there, right? Now this ovary is going to be converted into the fruit. Ovary is converted into fruit, whereas the ovule is going to be converted into the seed, and zygote into the embryo, and endosperm development is going to be formed. Okay, fine. So let us study about these things, guys, in detail. So this is called as a embryogeny. What do you mean by embryogeny? Yes, the zygote is formed, right? If I am going fast means kindly tell me I am not going fast. It is slow only. The zygote is formed, right? Now this zygote is going to undergo the mitosis. Okay. Now zygote is undergoing the mitosis and it is going to be developed into the embryo. Embryo. Okay. This formation of this embryo. Okay. Formation of embryo. Formation of embryo is called as embryogeny. Okay. The process of development of embryo from a zygote is called as embryogeny. Okay. So we have two types, right? One is a dicotyledon and the monocotyledon. In both, the early stages of the development of the embryo is similar. Remember this point. Okay. Fine. So the embryo is diploid in nature. Kindly make a note of this. Embryo is diploid in nature. Fine. Let us talk about the embryogeny in dicotyledon. Okay. I want everyone to listen here carefully. Okay. Now we are going to talk about the embryogeny. Embryogeny is nothing but development of the embryo. First, what will happen? Now we have a zygote, right? The one male gamete has fused with the zygote, right? Now this zygote is going to be divided mitotically. Means that is going to be divided unequally. Unequal. Okay. Unequally, it is going to be divided. Okay. When it is undergone the unequal divide, we have a two cell. Okay. One is a apical cell and another one is a basal cell. Okay. So remember here, this apical cell is going to this apical cell is at the chalazal end. Okay. And the basal cell is there. Both are divided unequally. Okay. And here you can see. Now, these two cells are there, right? One is a apical cell and another one is a basal cell. Now, this basal cell is going to be divided. Okay. This basal cell is going to be divided 6 to 8 times. 6 to 8 times. This basal cell is going to be divided 6 to 8 times and it is going to be form a suspensor. Remember, this is going to be called as suspensor. And this apical cell is going to be divided and it is going to give a fourth cell. This apical cell is nothing but the embryo. Okay. This mainly the embryo. Now it is divided. Now this embryo and that is attached to the suspensor cell. Right. This is 8 cells are there. Right. That is called as the suspensor cell. And this is mainly the embryo. Okay. Now, what is this function of suspensor cell? What is the function of this suspensor cell? This suspensor cell helping this embryo to push to the endosperm. To push to the endosperm. What is happening here mainly is the embryo is forming, right? 1 minute. First, what is happening here? The embryo is forming, right? Here, the formed embryo, it is going to divide and it's form the four cell, right? The suspensor cell first it is divided. What it will do is, it will push the It will push the embryo to the endosperm. It will push the endosperm. It will push the embryo to the endosperm. Why we require the embryo to go inside the endosperm? Because the endosperm is providing the nourishment and the food for the growing embryo, right? So that's why this suspensor cell is going to push. Okay, it is going to push this embryo. It is going to push the embryo to the endosperm. Okay, remember this. And after that, the stage that is will come is globular stage. Okay, remember this. The zygote will divide unequally to giving the two cell. One is the apical cell and another one is the basal cell. The apical cell is the embryo cell, whereas the basal cell is going to be divided into 6 to 8 times to give the suspensor cell. What is the function of suspensor cell? To pushing the embryo to the endosperm. Okay. Now, what is the next stage of the embryo is? The embryo is going to be divided continuously now. Now the embryo is going to form the globular stage. It will form like a globe-like structure. That is a globular stage. See here, globular stage. So now you can see here, there will be a stages. You can see here, guys. Egg, that egg is divided unequally to give the apical cell and the basal cell. From the apical cell, the embryo has been formed, right? And the embryo is after the formation of the suspensor, 6 to 8 cell suspensor. Now the cell is divided, the embryo is divided divided into the 4 cell. Now the stage is globular stage, then triangular stage. Now the stage. Why it is called as heart stage means it is forming the heart shape, right? So that's why it is calling as the heart shape. Then torpedo stage, then a mature embryo. Till there, the suspensor cell is there. You can observe here. Okay, now the mature embryo is formed. So this is the stages of the embryo. How can you remember GTH? GTH, TM. You have to remember these stages is important. Globular stage, triangular, heart stage, torpedo, and mature embryo. Okay. I think, do you understood this embryo development, guys? Do you understood this embryo development? Can you give me thumbs up or your emojis, message, any kind, anything? Very good, very good. Okay, I think you understood, right? Now let us go to the next. Fine. Okay, I think you understood this, guys. Now let us talk about, I already told you that the formation of the embryo is similar in the monocot and similar in the dicot. But the embryo, like after the embryo formation, that after the embryo is different, but the formation of the embryo stages is same in the both monocotyledon and dicotyledon. Okay, fine. Now let us talk about the dicotyledon embryo, guys. Okay, kindly see here. Okay, observe here. There is a dicotyledon embryo. Okay, we know that if it is a dicotyledon means it should have two cotyledons. Yes or no? Two cotyledons. Here you can see this is a one cotyledon, and here you can observe there is another cotyledon. Are you observing right, guys? See here, this is a one cotyledon and this is a second cotyledon. So these are called as a cotyledon. Okay. Have you seen, guys, when you divide the piece, or when you divide the chickpea, uh, exactly you will get exactly you will get in the two equal of. Yes or no? While eating the chickpea or while eating the peas, have you seen? If you cut the pea or if you cut the chickpea means equally you will get the two parts. Yes or no? How many of you seen? Two equal part. This two equal part you will see, right? If you open, you will see these two equal parts. Yes or no? Have you seen, guys? Groundnut. At least have you seen groundnut? Groundnut, when you equally two parts you will get, right? Where if all are in the home, kindly see once, okay? if you open the groundnut, it will become the two equal parts, right? Two equal parts, and you can easily see some a small, like a small white color structure will be there, right? A small white color structure will be there, right? That is nothing but embryonal axis. You can see that middle of that is going to be form a embryonal axis. Embryonal axis. Okay, fine. Here you can see. Okay, this part is going to call as the embryonal axis. Okay, the center part we are going to call as embryonal axis. Embryonal axis. Okay, I will written like EA. Okay, and here you have to see that above the cotyledon, here you can see the above the cotyledon, there is a part, the part which is above the cotyledon, we are calling as the epicotyle. Okay, the part above the cotyledon we are going to call as the epicotyle, and the part that is below the cotyledon, that is you can see here, the below the cotyledon, it is called as the hypocotyle. Okay, the epicotyle is going to give the shoot. The epicotyle part that is going to give the shoot, whereas the hypocotyle part that is going to give a root. That is the part below the cotyledon. The part below the cotyledon. Here I will write. Okay, kindly make a note of this. Epicotyle. Okay, epicotyle is above the cotyledon. The structure above the cotyledon. And this epicotyle will give rise to the shoot. Okay. Then coming to the hypocotyle here, hypocotyle below the cotyledon. Below cotyledon, and this will give rise to the root. This will give rise to the root, and this epicotyle will give rise to the shoot. Okay. Then you have seen that this is the embryonal axis, center wala, that is called as the embryonal axis. And here you can see below the below the hypocotyle, there is a radicel is there, right? Radicel is going to be from the root. This is going to be from the future root. Okay. And you know that there is a root that is covered by, here you can see this root is covered by the root cap. Do you understood the structure, guys? And here you can see plumule that is going to give a shoot. Plumule will give rise to the shoot, and radicel is going to give a root. Have you understood this structure? Can you give me thumbs up, or can you tell me in the chat box whether you understood or you want to repeat once again? You want me to repeat once again, Navya? Okay. Navya, you want me to repeat or you understood? Okay, thank you, Navya. I will repeat, guys. 1 minute. Yeah, I will repeat. See once again. Okay, see once again. I'll remove this, okay, because I will explain, That's why I will repeat this. I'll repeat once again, kindly see. Okay. See here, guys, we all know that dicotyledon embryo. Dicotyledon embryo means it is consisting of the two cotyledon, right? This is the one cotyledon. And here you can see this is a second cotyledon, right? This is a second cotyledon. Okay, this is forming the structure like this. Okay. This is a two cotyledon, and this is called as inside the center of this cotyledon, we are going to call as center of this cotyledon, we are going to call as embryonal axis. All the other parts are situated in this embryonal axis. Okay, embryonal axis means center part. Central. Okay, central of two cotyledon. Okay, I told you. If you break down the groundnut, or if you break down the pea, means it is going to get the equal part, right? In the center of this, there is a embryonal axis will be there. Okay. And the epicotyle, the structure above here the epicotyle is present above the cotyledon, right? Here you can see this is the cotyledon part, right? Till here is the cotyledon part, and here you can see this structure The structure is above the cotyledon. So that's why it is called as epicotyle. It is going to be from the shoot. Okay. Shoot part. Shoot part is going to be arise from the epicotyle. And plumule is above the epicotyle, that is going to form the shoot. Shoot part is by epicotyle. Okay. Whereas the plumule is going to be from the shoot part. Okay. Then coming to the, here you can coming to the hypocotyle. Hypocotyle, here you can see this is below the cotyledon. Below the cotyledon. So that's why it is below the cotyledon. Below the cotyledon. Okay. And this is going to be from the root structure. Root. Okay. Then coming to the radicel. This radicel here is, it is going to be from the root. Okay. And here it is covered by the root cap. Root is going to be covered by the root cap for the gravitation purpose. Okay. Root cap. This is the structure of dicotyledon embryo. Now you understood, guys? Can you give me thumbs up or can you give me any reply in the chat box? Nikita, now you understood? So this is nothing but the embryogeny in monocotyledon. It is similar like the. Okay. Yes. Thank you, Nikita. Thank you, Sushti. Thank you, Navya. Okay. So this is the embryogeny, means embryo formation in the monocotyledon. As I told you, it is a similar way. Like the zygote is going to be formed and it is going to be divided unequally. Unequally. And there is a basal cell and there will be a terminal cell. Basal cell and the terminal cell, you can see that the terminal cell is going to form the suspensor cell, right? The suspensor cell is going to be push the embryo. That is similar globular stage. Okay, now let us talk about the monocotyledon embryo. That is similar. That's why I didn't explain that monocotyledon embryogeny. Okay, fine. Now let us talk about the monocotyledon embryo. You all know that the monocotyledon is having the one embryo, one cotyledon or two cotyledon, guys. Monocotyledon is having one cotyledon or two cotyledon. The rice, if you take the rice is having one cotyledon, yes or no? Yes. Mono means one. Yes, Nikita. Very good, Navya. Very good, Jirmili. So that is one cotyledon, right? So this is the structure of the monocotyledon embryo. Let us study about this. Here you can see, guys. This, here you can see, this part is called as a cotyledon. See here, this part is called as a cotyledon. Cotyledon, and this cotyledon is located laterally. Laterally. Laterally means if you are sitting, right? Where is your limbs are there, guys? Where is your limbs are located? Either in the center or in the lateral stage. Can you tell me where is your hand and leg is located? Laterally or in the center? Your face is located at the center, right? Whereas the hand and the legs are located. Where is your hands and legs are located, guys? Heads are located centrally, whereas the legs and hands are located laterally, right? Lateral position this is, right? Here also, in the monocotyledon, the cotyledon is located at the lateral. Okay. Yeah. So this is called as the scutellum. 1 minute. Yeah. So this part is called as the scutellum. There is a question will arise to you. Okay. The question is the cotyledon in the grass family or the cotyledon in the monocotyledons called as what? Scutellum. Okay. Remember, it is called as scutellum. It is located laterally. Okay. So now here you can see that structure. Okay. The structure is coleoptile. So this coleoptile we are going to be called as, it is going to be, coleoptile is the structure that is going to present here. You can see this is a structure, coleoptile. This is going to be cover the shoot. It is going to be cover the shoot. I think you have studied about the coleoptile. In the canary grass, in the which chapter you have studied, guys? In the 11th, you have studied about the coleoptile. Yes or no? Coleoptile, where you studied? Very good, JNV Hyderabad. Very good, Nikita. In which chapter you studied coleoptile, guys? Chapter name? Can you go back to the 11th? Plant growth and development. Plant growth regulators. In that, the first hormone. Which is that hormone? Which hormone is identified in the coleoptile? Can anyone tell me which hormone is identified in the coleoptile? Come on, guys. So you forget about the 11th syllabus. Very good, Tanuja. Very good, Venu. Our students are brilliant, but they're very lazy to type in the chat box. Yes, Bijay. Very good. Very good. Maybe auxin. Ranganaiki, correct. That is auxin only. Yes, Nikita, auxin. Yeah. The coleoptile is nothing but it is covering the shoot. Okay. It is covering the shoot. Phototropism will be happen, right? Yeah. Very good. Okay. Okay. It is going to be cover the shoot and then coming to the shoot apex. Here you can see, guys. This is nothing but the shoot apex. Okay. This is the shoot apex. Okay. And this is nothing but the coleoptile. The coleoptile is covering the shoot apex. Okay. And then coming to the radicel. You all know that what is the radicel? Radicel is nothing but the root. It is going to give a future root, right? And then coming to the coleorhiza. What is the coleorhiza? Coleorhiza is going to be cover the root. It is going to be cover root. Coleorhiza cover the root, whereas the coleoptile will going to cover the shoot. Do you understood, guys? Coleorhiza is going to be cover the root, whereas the coleoptile will cover the shoot. Do you understood, guys? Then we are left out with the epiblast. We left out with the epiblast. What do you mean by epiblast is it is a rudimentary. It is rudimentary structure. Rudimentary structure. Okay. Rudimentary cotyledon. Rudimentary cotyledon. Rudimentary structure of the cotyledon. This is about the monocotyledon embryo. Okay, I will explain you once again, guys. Kindly look at this once again. This is the cotyledon. That cotyledon is located laterally. Okay, this is called as the scutellum also. Then coming to the first, this is called as the cone-like structure. That is called as a coleoptile. That is going to be cover the inner shoot. You can see here that is going to be cover the shoot. Then coming to the root, that is the terminal. Terminally, there is a radicel is there. That radicel is going to give a future root. That future root is going to be covered by a coleorhiza. Coleorhiza will cover the root. It is as simple as that. Very, very simple and easy. Do you understood that? What do you mean by rudimentary structure, guys? What do you mean by rudimentary structure? Which is the rudimentary structure present in the human? Which is the rudimentary structure? And I have a one question, guys. During the evolution, which is came first? Either monocotyledon or dicotyledon? Very good, Venu. Appendix, very good. During the evolution, guys, which one came first? Dicotyledon arises first or the monocotyledon arises first? Which one is arises? Monocotyledon or dicotyledon? Yes, everyone telling mono because mono means what? One. One means first. It is first arising. That's why it is monocotyledon will come first. I have a question for you. Which is the root type in the monocotyledon? Fibrous or fibrous root or taproot? Very good. Tailbone. Very good. Hyderabad. Tailbone. Very good. Yes, Venu. Fibrous. Money is also telling fibrous. Very good, fibrous. Then what about the root that is present in the gymnosperm? In the gymnosperm, what type of the root you can see? In the gymnosperm? If anyone know, tell me. Gymnosperm. Gymnosperm will come before the angiosperm, right? In the gymnosperm, what type of the root is there? Taproot. Taproot. Very good, Tanuja. Very good, Venu. In the gymnosperm, taproot is there. After the gymnosperm, now you tell me, monocotyledon came first or dicotyledon came first? Now you tell me which one came first, monocotyledon or dicotyledon? Dicotyledon. Just compare the root, guys. Monocotyledon is having the fibrous root that is arises after that. So that's why there is another embryo should be there, right? Another cotyledon should be there, right? That cotyledon become rudimentary here. How the before we are having the appendix, it is working. Now it has become rudimentary, right? Like that, there is another cotyledon there before. Now this become monocotyledon. The another cotyledon become rudimentary here. That structure we are going to call as epiblast. Epiblast. You can see there epiblast here. Okay, now you understood which is came first, dicotyledon, not monocotyledon. Okay, yeah. Okay, guys. Today we completed with the double fertilization and also the post fertilization, that is the embryo formation and the monocotyledon embryo and the dicotyledon embryo. I assume that you understood these two topics. These are very important in case diagram and the labeling also. The homework for you is you have to practice the both diagram, guys. Okay, you have to practice both the diagram, monocotyledon diagram and the dicotyledon diagram, and you have to label by yourself. Okay, you have to label by yourself without seeing the diagram, without seeing the labeling, and by only you have to write the diagram without looking anywhere, and you have to label the diagram. Okay, this is homework for you. Fine. Let us see how many will you do this homework in the next class. Okay, Mahalakshmi, I will tell you rudimentary means. See here, we all are humans, right? We all are humans. The organs, once upon a time, it is working. Okay, 1 minute. 1 minute, I will tell you. Give me a time. Okay. So see here, the organ is there, okay? That organ, once upon a time, it is working. For example, you can see the ancestor of ours, like they are eating they are eating the fruits and the vegetables without boiling, right? Or raw vegetables they are eating, right? Mahalakshmi, yes or no? Our ancestors are eating the raw vegetables, right? Here, in the appendix, the raw vegetables are these are having the enzyme, the appendix having some enzyme that is used to release the enzyme that is going to break down the raw vegetables and the whichever the thing we eaten raw, that will be broken down by this appendix enzyme. Sorry. Okay. But now what happened? We are eating any raw vegetables and all the thing. We are boiling and we are eating, right? That's why now it become rudimentary. It is not working now. Okay. That organ, it is working once upon a time. Now it is not working. That is called as a rudimentary. For example, tailbone also. The ancestors having the tailbone, right? Now we are not having the tail, but tailbone is there. Okay. Very good, Ranganaiki. Sorry, guys. Ranganaiki is Ranganaiki is asking the very good question. That is, yeah, Ranganaiki asked the very good question. That is, Yeah, that is, ma'am, when the mango stamens or the anther or the pollen grain falls on the guava flower. Okay. So is there any chance of combining of a new plant? Ranganaike is asking the question. Now I want answer from the students yourself only. Answer me for the Ranganaike question, guys. I will answer. Is there any chance of combining and forming a new plant? Ranganaike, your friends will answer now. Malakshmi is telling yes. What about others? Nikita is telling no. And Money is also have a question, that is, ma'am, which one is more organized? Whether the monocot or the dicotyledon? Monocotyledon and dicotyledon, if we compare, means both are organized only, but they are different types. That's it. Very good, Venu. Very good. Tanuja is also told no, and Nikita is also told no, and Venu also told no. Why? Because it's incompatible. As we studied the yesterday, one of the student asked the question about the mustard flower. Mustard flower, the in the own flower, the pollen grain is incompatible. So here also what will happen? It is a different species, right? If it is a different species means the pollen grain will be not germinate on the stigma, right? So that's why it will become incompatible. Okay? It will become incompatible, ma. Okay. Okay, Ranganaik. Fine. Thank you, students. All our answer to this question. Fine. So let us meet in the tomorrow's class. I think you understood. I will go for the poll, guys. You can give me your feedback in the poll, okay? Yeah, I have launched the feedback. And one more thing, guys. If you want to understood any topic, means what you have to do is you have to read once that topic. If I told you in the class also, if any teachers teach you in the class also, what you have to do, the final thing is you, right? You have to read that concept once again. Okay. And you have to discuss the topic with your friends like a story form. Then only it will become a understandable and it will be easy. Okay. Whenever you're going to mess or whenever you're going to for the walking, right, just explain the topic to your close friend or explain the topic to anyone who didn't understood, then it will become very easy, right? Nothing will be difficult, guys. Nothing will be difficult. Okay, understood this thing? Fine. Thank you so much. Let us meet in the next class. Thank you, Mahalakshmi. Bye, Monica. Bye, Nikita. Ranganaiki personal notes are not there, pa. We will give you mind map and this PPT we will provide you. Okay. And Avanti modules also there, right? We will teach from the modules only. And whenever the topic is completed, no, kindly solve the modules question, guys. When you solve the module question, you will get the question. So many questions that also you can ask the questions in the class. Okay, thank you, guys. Let us meet in the next class.